Good morning, all. Welcome.
Good morning, everyone. Good to see you all. We'll just give it a minute to let some people in. We'll get started. Thanks for joining. Great. Well, welcome everyone. I think people will still be signing on, but we'll go ahead and get started. It's great to see you all. I hope you're having a good morning, afternoon, or evening, wherever you're joining us from. I'm Amy Byrne. I am at the Morton Arboretum in the States, and we lead the Global Conservation Consortium for Oak, which we are so happy to support and co-host these webinars with the International Oak Society. As you'll see on my screen, this is our 9th webinar, which is amazing. Thank you to all our past and current and future presenters who join us for these webinars. So we just like to recognize as well the supporting institutional and standard institutional members, which you'll see on my screen here as well. Just, supporting the iOS mission and goals and objectives through their support. So we just like to recognize them. Thank you all, and thank you to all our members as well. And so yeah, today for the 9th webinar, we have two wonderful presentations. Dr. Sandra Denman will be talking about acute oak decline, and then Dr. Yigang Song will be telling us a bit more about the Shanghai Chenshan Botanical Garden oak collections. But I'm going to pass it off to Roderick now, with the president of the International Oak Society, who will have some announcements for us.
Thank you, Amy, and greetings, everyone. Thank you for joining us today. So I think you may have seen, or I hope you've seen the latest newsletter. And basically, I want to go through some announcements that are there in case you haven't read it. As you know, we got the conference coming up in Oaxaca in October. And at the conference, we'll have our triennial meeting of members. And this time, we're actually going to try and open that up to virtual participants as well. So you will be receiving, I hope, if we can make this work, a link where if you're not able to join us at the conference, you may be able to join us for that particular meeting, where we have a review of what's happened in the last 3 years, what we're planning for the next 3 years, and we will also choose our new board. I say choose, but in fact, what happens is there are candidates, and if the number of candidates is smaller than the total allowed candidates, then basically they're all in. So we ask members to confirm the candidates. I'm going to share a link in the chat, if I get this right, to the from the board, and that has links to the candidates, the candidate statements, and especially for the new board candidates, very happy to welcome 4 new candidates to the board, so you can read about them there. Also, we've published our financial reports. If you're interested in that, you can look those up. There's links in that article I just shared, in that post. I think that is all I had to announce. I just want to say that I don't know, Amy, if this is right, but we haven't yet been able to connect with Yigang Song.
He just joined. We're good.
Great.
Yeah.
Excellent. So very happy to have our 2 presentations today. I had the pleasure of meeting Sandra at Forestry Research in UK this year, and she actually showed me in her lab what she's going to talk about today, and I thought it'd be very good material to share with members today, and also very keen to learn about the collection at Shanghai Chenshan Botanical Garden, which is a new collection which promises to be very impressive indeed. So with that, please remember that at the end, we'll have a Q&A after each presentation. And then after the end of the presentations, we'll open it up for any general questions about the iOS. So if you have any of those, any about the meeting of members or about the conference, please hold them till the end. Okay, so I think if we're ready, we can go to Sandra's presentation. I will be sharing her presentation now. Let's see if we can get this right. Let me know. Hang on. No, wait.
Not here yet, Roderick.
You're not there?
It takes
Okay,
a
just
little
one
time
second.
to load, I think. It's usually
Hang
quite
on
a big file. Yeah.
Share.
Looking good. Something's happening. Yep, we're good.
Okay.
Okay.
Floor is yours.
Right, well, great. Okay. Well, thank you very much, Roderick and Amy and all of you. It's lovely to be here today. In the UK, it's especially lovely because we've got a little bit of rain. And I know we fondly refer to as Mud Island, but we've certainly been a dry cake island this year. We've had a dreadful drought, so we really are very pleased for the rain and grateful indeed. Anyway, the way we're going to conduct this is Roderick's going to change the slides because we've got electric works going on in the laboratory today, and I was just a bit nervous that it would all fail on you. So what I'm really going to talk about is changed behaviours that have a damaging outcome. And I'm going to sort of root it all around the acute oak decline research that we've been doing, where we explore the environment and the host, pest, and microbe, really pathogen relationships. So could I have the next slide, please, Roderick? Thank you. So just as a bit of background, oak declines are really complex, very complex syndromes that cause the overall deterioration in the health of oak trees and usually lead ultimately to their death. Because it's so complex, it's been quite difficult to sort of unravel and unpick. And in the UK, what we have found is that we've got two sort of different types of oak declines, which are really part of the same continuum, if you like. But just for ease of trying to investigate them, we've separated them out into different categories. And so these oak declines, the one is called chronic oak decline, which, as its name suggests, it takes place over a long time. And actually, the photograph that you're looking at is a picture of a tree that is suffering from chronic oak decline, where there's a lot of fine branch shedding, the branches look stubby, the leaves yellow, and the tree takes a long time to die. Now, usually that's driven by, first of all, environmental factors, but from a pathogen point of view, there's a malfunctioning of roots. And these can be the big buttress roots and or the feeder roots. But what we're going to focus on today is on acute oak decline. And the relationship between acute oak decline and chronic oak decline, that could be happen together, that they happen together, or the one could follow the other as part of a continuum. But for order or ease of understanding the whole complex, we've divided them into two separate categories, and I will be talking about acute oak decline. The point I want to make about all decline diseases is that they're underpinned by the weakening of the trees in the first instance. Could I have the next slide, please? So to understand how trees become weakened, here's a very simple model. Trees need water, nutrients, and indeed sunlight to grow. And if anything impedes the uptake of water or nutrients, the trees will respond. And they will respond to these shortages and deficiencies, and usually imbalances occur in the tree, and the cost to the tree is their health status. So trees become stressed or weakened, and then there's an increased risk of succumbing to pests and diseases. Now, this weakening affects the normal functioning of the tree. They become less resilient. And in the case of acute oak decline, we've developed a more complex model. Could I have the next slide, please? So we work on this model, which was originally thought about and conceived by a very famous forest pathologist called Paul Magnon, and it was improved by Magnon and Lachance in 1991. We have updated that model, bringing in a lot of other factors. So the way that predisposition works then is that in the first stage, that's near that pink arrow that you see in the spiral diagram, you can see it says predisposition. And in this first stage, usually it's environmental factors that are the key stressors, and these can be things like poor soils, such as soil nutrient deficiencies or pH imbalances, compactions, flooding, fluctuating water tables. It can also involve climatic factors, such as severe drought or heat waves. And these can occur concurrently or alternately with some major biotic factors such as insect defoliation or even primary diseases. One that we all know about is the mildew that affects trees, particularly across Europe and in the UK. And so these weakening or predisposing factors work on the trees. The microbial communities, their microbiomes become affected. The soil pH balance affects the soil, and we get this total sort of imbalance within the whole system. And sometimes human activity adds to that too. For example, if people have overstocked or even stocked land, if trees are too densely planted and not thinned timely, or even social use, such as increased footfall, putting in of pathways, water abstraction, that kind of thing. These will all stress the trees and reduce their health and defenses. And then it will follow into the second phase, which is called the inciting phase, and that's really a tipping phase. And in a tipping phase, you can have a single or sequential major events like major droughts or major wind events that go sustained, floods even, things like that. That tips the trees sort of beyond a point of no return, as it were, in terms of pathogens. And that takes us down into the contributing phase where the microbes and insects and that, which are really secondary. These are not the primary pathogens. These are mostly the secondary pathogens. These pathogens and pests start to be able to attack the trees, whereas previously they couldn't do so. They had to wait until the trees were stressed and their defenses were reduced. And so the tree will spiral down into this position. Sometimes what happens is that it's only part of a tree, or in fact, it's a passing phase in the tree, and the trees are able to sort of come out of that. But in our experience with AOD, even if they go into slight remission, we always find that actually the end point is death. It's almost like it's just delaying the process even longer. So if I could have the next slide, I'll show you what the symptoms of AOD look like. So looking at the trees, these are on native trees to the UK. So they're temperate oak species, Quercus robur and Quercus petraea. And what one would see is on the stems or the trunks of the trees, these weeping patches. And these weeping patches have got a sort of vertical arrangement to them. And the weep actually emanates from a crack between the bark plates. You can see that in the fourth image from the left-hand side. Now, underneath those weeping patches, you would find that the tissue, the live bark, in other words, the conducting vascular tissues of the tree, is becoming necrotic. And if you have a look at the weeping patches and the photograph next to it in the third panel, you will see that the weeping patches approximate to those necrotic patches. Now, those necrotic patches are caused by a pathobiome of bacteria, in which one of the most damaging species is Brunneria goodwinii. In the lower series of panels, the second group shows you a cross-section through bark that's been infected, and you can see that actually that necrosis actually macerates the tissue, creating a cavity. And so this interrupts the flow of water and nutrients from the roots to the crown and from the leaves, obviously, down to the roots. And so the trees start to go into starvation and indeed water shortage. Now, coming back to the external symptoms, in many trees you would see the D-shaped exit holes of a little native beetle called the oak jewel beetle, Agrillus bigotatus. And we don't always see those exit holes because the larvae themselves actually are quite vulnerable, and they don't always complete their life cycle. However, if you open the bark, pull away the outer bark and go into the inner bark, right onto that cambial interface, as you see in the very bottom right-hand corner, in AOD, you will always see the co-occurrence of the galleries of the larval stage of Agrillus bigotatus and the necrotic patches that are caused by the bacteria. So the external symptoms are fairly diagnostic, especially once your eye is trained, although many other pathogens, for example, armillaria or phytophthora, that can also cause bleeding, but they're fairly diagnostic. But it's once you go into that tissue that it really is confirmed. Now, we've shown in the UK that the bacteria are responsible for this. I already talked to you about Brunneria goodwinii. There are other bacterial species, Gibsella quercinecans and Lonsdale britannica, that are also involved. But the key one, the most damaging one, is Brunneria goodwinii. Now, the larval galleries, as we say, are always associated with it. Could I have the next slide, please, Roderick? But the role of this beetle is difficult to elucidate, and it's been really quite challenging to prove it. But this role, because I think the beetle is native, it's always been quite emotive and quite controversial. And that's also because in the UK, this beetle was listed as rare and in danger in the 1987 Red Data Book. Now, some of the reasons for that is that it's quite difficult to see. The adults are only 13 millimeters long. They emerge around about June time. They live for about 8 to 10 weeks. And once they're fed and mated, the females lay their eggs in those vertical cracks, not under the bark plates, but they lay them in those cracks. And the larvae then burrow into the live tissues where they live out of sight for 18 to 22 months. Larvae cannot ingress into healthy tissues, but they need to have weakened trees. And the galleries also girdle the trees, cutting off further tree water and nutrient supply. Now, I mentioned that it's been quite controversial. The big argument was that there was no evidence to support a causal role for the beetle, and so we began that journey. Could I have the next slide, please? So I had a wonderful MSc student who then became a PhD student with me and is now still with the project 15 years on. We're waiting for the next slide, Roderick, if it can come.
Oh, sorry.
Okay, don't worry.
I think I was just cut off recently. Anyway, I'm back now.
Oh, sorry. Yeah, there we go. That's perfect. Thank you. And this student, Nathan Brown, he got hold of the forestry research records and the distribution of AOD, and also looked at the conservation records from the National Biodiversity Network, and the two fitted hand in hand. And so that was already quite a good starting point, that there was really a sort of a dependent relationship between the two, or causal relationship. Next slide, please. And so Nathan, together with our biogeochemist Elena Vangulova, they carried out a desktop study where we used survey data and all the records that we had, as well as tapped into all the existing spatial data sets, so different climatic, temperature, atmospheric distribution, maps, soil maps, ecological classifications. And they did a desktop study to look for correlations. And what they found was that AOD occurred in warmer areas in the UK, in low rainfall areas, and the warmer areas tended to be at low elevation, so I think that wasn't really a big surprise. But where there were high nitrous oxides in deposition, particularly as dry nitrogen on the surface of the leaves, but also if it rains through as wet nitrogen, and low sulfur deposition and on droughted soils. Now, all of these conditions really suit the beetles. Could I have the next slide, please? And the reason that they suit the beetles is because beetles like warm, sunny conditions. They don't like cold, wet conditions to survive. And there you can see the AOD areas were found where there's much higher temperatures, also much lower rainfall, as much as 200 millimeters a year difference where they're found. And these places generally tend to be in the east, the southeast, and the Midlands of the UK. Occasionally, or we did find it along the Welsh borders. And as the climate has changed over the 15 years that we've been looking for that, we found that it's actually spread a little bit more to the Welsh borders and further northerly, sort of playing to the whole climate change driver. Also, what was really interesting was the high dry nitrogen deposition. Now, the nitrogen falls from the atmosphere onto the surface of the leaves, and then leaves actually become more attractive to insects to eat, because nitrogen is one of the key building blocks of proteins in insect bodies. And so they really start to munch the leaves a lot more. So the adult stage of the beetle is attracted to these areas as well. And interestingly, in the 1970s in the UK, there was quite a lot of concern about air pollution and acid rain. And so a lot of factory emissions, there was quite a lot of control with that. And along with that came a decline in sulfur emissions. Now, sulfur is sulfur is a bit of an antimicrobial agent. And so it's interesting that AOD has occurred where we've also got this low sulfur levels. Could I have the next slide, please? So, oops, one back, thank you. Yeah. So we had already done quite a lot of work on log tests and with the bacteria to name them. We had tested them in sawn off logs from oak trees, put the bacteria in, and we could get lesions, but I have to confess it was a struggle. The bacteria didn't seem to want to do it altogether, as one would expect with a primary pathogen. And so we kept on thinking about this relationship with the Agrillus larvae. So we also learned how to capture the beetles, rear them in captivity, which is what Roderick was telling you. He saw a little bit about when he was at our laboratories. And so we learned how to rear them in captivity, get them to mate and lay eggs. And then we could put the eggs together with the bacteria onto the logs. And what we found was that we got significantly bigger lesions when we put the two of them together. And then we had a very talented PhD student, James Doonan, who worked together with my work partner, James MacDonald, and another postdoc, Martin Broberg. And they looked at the transcriptomic analysis of these interactions in the log tests, and they found that Brunneria goodwinii seemed to upregulate a lot of pathogenicity genes in the presence of the larvae. So this was really starting to move towards, getting really good evidence that there was something going on between the larvae and the bacteria. But we needed to prove this in the field. So could we have next slide, please? And so I've talked to you previously about predisposition, and that's really an essential part, a driver of AOD. And so the question we wanted to ask is, if we droughted trees, would AOD and Agrillus ingress into the tree? And so we created a field trial where we had 3 environmental treatments. We had drought, where we constructed rain exclusion shelters around our trees. We ring-barked trees almost fully as a proxy for nutrient and water deficiency, and then our control trees were just ambient. Then we applied our pathology treatment, so our control trees had no disease agents. We put only bacteria or only Agrillus eggs or a combination of them. And we did apply them in 2 ways, but the non-wound way is the way I'll be talking about today, because that most approximates the situation in nature. You can see we had 6 block replications, 144 trees, 576 data points. So it was a big job. So could I have the next slide, please? So on the left-hand side of the screen, you can see what happened. The green curve is the control trees, and the purple curve is the droughted trees. And that's just an indication that our soil moisture levels were working. Sorry, it's the soil moisture levels. So the controls are the green and the purple is the drought. And you can see it's the time span there is from July 2021, and each little graph shows a different depth of the soil, so 10, 30, and 50 centimeters deep, and it runs until about September 2022. Now, some of you will notice that the green curve meets the purple curve in about July 2022. And what happened was, unfortunately, we had a great big natural drought in 2022 in the UK. And therein is, a story about wanting to prove things in nature. It doesn't always work within 1 or 2 years. You've sometimes got to do more sustained trials. So the good thing about that was we could see that actually our drought shelters worked, and that the levels attained there were what would happen in reality. So we knew that was working. And you could also see that the effect of the droughting was quite cumulative. So in the graph below there, you can see that the effect between on growth, so that the second graph is on tree growth. You can see that after 3 months, there was a small difference, but after 12 months, there was quite a large difference. So that wasn't the only sort of, should I say, challenging thing that we got in the field trials. Some of the results were a little bit unexpected as well. So what we did find was that all of the control treatments, so whether the trees were droughted or environmental control or ring barked, the bark was all still pretty healthy. There was no lesions, there were no weeping, that was all fine. And actually we found the same when we applied the bacteria only from the outside. We got no real impact going on. So what we had expected was, if we put together the bacteria and Agrillus. So you're seeing the 4 little panels, graph panels up in the top right-hand corner, and you're looking at the bottom one of that, the non-wound both. So we had expected that when we would apply both of these, we had expected to see the most severe AOD and indeed to back isolate the bacteria. And surprise, surprise, what we found is that while we did get that and we did get lesions that developed, we found really that if you look to the left of that, you'll see a series of 3 panels. When we applied Agrillus only, so only the eggs of the beetle, we actually got the worst AOD, particularly in the ring-barked trees, and we could back isolate all of the bacteria. So this was really quite an interesting thing. We wondered whether the eggs had transmitted the bacteria or what had happened. What also did happen and shouldn't have happened was unexpected. If you have a look in those non-wound Agrillus inoculations, we hadn't expected anything to happen in the control trees. There we are. Go back one, Roger, just back one. Back one, that's it. Yep, to that one. We hadn't expected any ingress to happen. But actually, Agrillus could get in. But it didn't go anywhere. It just started to make a larval gallery, and then the larvae died. And I think that probably, that ties very much to the drought that we had in 2022, that even the drought was so serious, the natural drought was so serious that even the control trees became susceptible to ingress. But there was no question about it that the trees that had been ring-barked were the most vulnerable. So the trees that had depleted carbohydrates and water status, but also the drought trees, and you can see there from the lesions, they were the ones that got the worst affected. So we did answer the question, does drought predisposition affect the colonization and disease severity? Yes, it does. Does the ring barking make it more severe? Yes, it does. Can AOD form in the absence of Agrillus? No, we couldn't get that. See the 2 top panels, we couldn't get that. And is AOD necessary for development? Yes, it is. So another unexpected result was that in those control trees, we isolated Brunneria goodwinii from about 10% of the non-symptomatic control trees, suggesting that Brunneria goodwinii exists in any event in the trees as an endophyte. Next slide, please. And so we conducted an experiment. We wanted to just investigate that interaction between the beetle and the larvae a little bit more. And so what we did in the lab, we produced larvae and we harvested them. We gave those larvae to our chemical ecologists from Rothamsted Research. What they did was extract chemicals from the bodies of the larvae. They did analysis on them and they separated out 6 fractions, were sort of a mixture with chemicals. Each one of those fractions was tested separately by adding it to a test tube with bacterial culture, and we measured 2 things. We measured the growth rate of the bacteria and we measured the gene expression of the bacteria. And that was done by Maureen Cambon and James MacDonald at Birmingham University. Next slide, please. What we found was amazing. It's actually been quite transformational. What we found was that one of the 6 fractions that we tested caused the bacteria to grow and multiply more quickly than the control. So you can see that in the 3 top panels. You can see we had 4 or 5 replicates where each one, the yellow curves are where we've got the larval extract, and you're looking at the growth rate going from left to right is speeded up, and you can see that there were significant differences. When Maureen did the transcriptomics on it, what we found is that over 400 pathogenicity genes were upregulated in the presence of those larval extracts. And so what we've seen is that these chemicals off the body of the larvae cause a change in the behavior of the bacteria, triggering those pathogenicity genes. Now, more work does need to be done, but that's been pretty transformational. Can I have the next slide, please, Roderick? So just to sort of sum up, I've tried to show you that we've been looking at changes in situations. So changes at the landscape scale, where we've had climate change driving drought and heat wave, various anthropic behaviors driving air, soil, water pollutants, soil compactions. The results of these is that they result in things like increased insect herbivory, soil nutrient imbalances, microbial imbalances, and all of these change the tree's functions. The drought and heat waves can cause embolisms in the tree's vascular systems. The stomata close, the trees go into hydraulic failure and carbon depletion or starvation. All of these things suit the insect, and so the trees weaken, and the insect can lay the eggs on it. The larvae come into contact with the bacteria turn nasty, you get lesions, and then the oak trees die. I think I'm just about there now. Roderick, could I have the next slide? And so it's not all doom and gloom. What can we do to help prevent AOD? We can avoid predisposing our oak trees, avoid compaction, or if there's a necessity for compaction, try to decompact after compaction. Avoid wounding the trees. Look out, be careful about putting stock on lands. Too many, the fecal deposits, the urine damage, all of this is not good, is not helping our oak trees. Next slide, please. And so I think that the whole thing is that we've got to relieve predisposition in a naturalistic way in tree time, not to hurry it. Trees don't respond to sudden change well. As oak lovers, all of us are quercophiles, we need to stay informed. We need to be engaged and participate. I invite you to join our Oak Hope group. Please feel free to contact Sally Simpson. Our Oak Hope group is a network that we're trying to establish to help each other, help our oak trees, and to prevent and protect trees for future generations. My last slide, thanks, Roderick. There's so many, many people that I have to thank for this work. All my colleagues that have helped contribute to this, and our stakeholders, and to all of you. Thank you for listening, and I do hope to see you in Oak Hope at some time. Thank you.
Thank you very much, Sandra. That was fascinating. Rather depressing subject, but brilliant to see the research that's going on. And we do have some questions in the chat. Let me see if I can relay them now. Steve Potter asked, I'm not sure the question hasn't been answered already, but he's asked, are you aware of any cases of AOD in England in which Agaricus bigotatus was not implicated? And if so, were any alternative vectors identified?
It's a really good question, I have to say. The straight answer to that is no. Agaricus bigotatus is always involved, but sadly for us, we found that there are a number of new Agaricus species entering the UK, like Agaricus olivicola, and certainly in some areas there's a lot of Agaricus salsicollis that's come in. Now, Agaricus salsicollis is supposedly colonizes the upper parts of the tree, so more the branches rather than the stems. And I have a colleague who's a forester in Switzerland. He's been doing some trapping in their forests, and he sent me some beetles that they trapped, and amongst them was Agaricus salsicollis. And unfortunately, we have isolated the bacteria off that Agaricus salsicollis. So we do know there are more and more reports of AOD coming forward from Europe. And there is a chance that there could be Agrillus salsicollis involved in the whole complex, but we would need to find out whether those chemical compounds were produced by any other, indeed, insect species, not only Agrillus bigotatus, but at the moment it looks like it's only of Agrillus bigotatus.
Great. I think we have time for one more question before we move on to the second presentation. Jeroen Brackman asks, he says, is it like Dutch elm disease, associated fungus with a beetle? And then also a couple of follow-ups is, does climate change accelerate symptoms? And how can I detect this in the field?
Yes, okay. So there are some similarities with Dutch elm disease in that it's a complex involving both insect and microbes. It's different from Dutch elm disease in that Dutch elm disease is a fungus. So it's Ophiostoma ulmi and Nova ulmi that cause that. The fungus actually grows in the trees and blocks the vascular system, causing a drought within the trees because the vascular system is blocked. So there are some similarities, but it's slightly different in that Nova, the pathogen, so the bacteria in AOD seem to exist in the trees already, but in a benign form. They're just endophytes. They're minding their own business, it seems. And it seems as if there's been really an accident of nature that the two have come together. And that in itself becomes an interesting question, because have these bacteria always been there, or have they only recently sort of got into oak trees? We know that they come in other tree species as well. And the other thing is, have they changed? So the bacteria themselves are able to pick up genes through horizontal gene transfer. And so they could have, by mistake, picked up a lot of genes, which is causing them to become more pathogenic or have the potential to be pathogenic. So there's lots of interesting things there. In terms of climate change, the answer to that, the short answer is yes, because these heat waves and droughts and the stress factors are driving these diseases.
Great. Well, unfortunately, we do not have time for more questions, but do have a look in the chat, Sandra. Aside from all the comments saying how much they enjoyed your great presentation, there were a couple of questions. One from Khawla El Khatib from Morocco. It's quite interesting.
Wow,
Perhaps
amazing.
Perhaps we could also, people could contact you over email if you want to put your email in the chat if they have further
No
questions.
problem. We have people from North Africa in Oak Hope, and so we know that
great.
it's a problem in North Africa too. Thank you all for listening. It's been wonderful. Thank you, Roderick, for giving me the opportunity.
Sure. Thank you, Sandra. Moving on now to Yi Gang. Yi Gang, if you could unmute and share your screen, let me know.
Now it's okay.
Yeah, we can hear you.
I need to share my presentation, right?
Yeah, share your screen.
Oh, screen.
Okay. We don't see it yet, but it seems to be loading. Okay, now you need to go into presentation mode. We see your screen, but you're not in presentation mode yet. You go.
Okay.
There you go.
There is no presentation?
No, now you're all set. Take it away. Oh, now we're back to your
Okay,
screen.
I think my PDF version, maybe the PowerPoint version is too big.
Oh, okay.
Is it now?
We see it, but it's not presentation mode, not full screen. We do see your slide if you want to go ahead with that.
How about now? Is it okay?
Not yet. Yes, yes. There you go. Perfect.
Yes.
Good.
Okay. Sorry. Can I start?
Yes, please.
Okay. Good morning, good afternoon, or good evening, everybody. I'm Yi Gangsong, come from Shanghai Chenshan Botanical Garden. Today, I want to give a short presentation about the oaks collection in Shanghai Chenshan Botanical Garden, based on our research and the culture. I started working in Shanghai Chenshan Botanical Garden from 2012. Up to now, it's about 13 years. I working on the plant systematics and evolution group. From that time, I fell in love with oaks. Here is a full photo about our botanical garden from the autumn, spring and winter. And come to the oaks in China. Here is a map about the species diversity of oaks in China. From this map, we can see the high species diversity area is along the several mountains, like the southern part of Tibet area, the northwest of Yunnan province. Another part is the around the Sichuan Basin area. Around the Sichuan Basin area, there is a lot of mountain area. Is with high species diversity. Another part is between the boundary of Hunan province and the Guangdong province. There is a mountain line is the Nanling Mountain. The other area is along the East China area. Along here is also another Wuyi Mountain area. In China, we have three sections of oaks. All of them, not all of them, just one section from the New World oaks. The other three section is from the Old World oaks. The first one is the section Tachylabalanopsis, only distributed in East Asia and Southeast Asia. Um, there is about 90 to 120 species in this section. The main character is about the capula is a cycle, is not other is the cycle shape. Another section in China, main oaks, is about section Ilex. This section is distributed in the Eurasia area. In China, we have about more than 20 species. Another is we only have three species. This is also the Eurasia species, section series. In China, we have three species with very similar morphology. Um, in our botanical garden, we collected the oaks based on our research. First is about the acorn trait. As we know, the most famous trait is about the elongated cotyledonary petal. Um, in the whole oaks file based on the phylogeny tree of Andrew Professor Heap, uh, we found there are three linkages with this kind of ECP acorn traits. The first linkage is about is from the section Tachylabalanopsis. The linkage, the CTB linkage. Another one is the in section Ilex from the Himalaya, the Tibet area. This section Ilex species. And the last part is about the section is from the southern part of USA and Mexico area. Is this small these three section we now we know in the last section have this kind of of trait. All the other species with this normal cotyledonary petal. Based on our research, we found uh, sorry, we found the ECP is a divided trait, but this trait is not good for the speciation. Now in our botanical garden, we have collected all these all the traits from the different linkages. The first part is from the section Teclabonomosis, the CTB linkage. We have collected the seeds about Crocus rex. This is one of the most biggest acorns in the world, I think. We have working on the seed germination, and after germination, we receive the seedling in our botanical garden in the greenhouse because this species is from the tropical area. Shanghai is in the subtropical area. It's difficult to live in the outdoor area. Another one is Crocus twinkie, also from the CTB ingots. They also have the ECP acorn traits. For this species, because this species is from the East Asia, East China area, so it is suitable for outdoor growth in Shanghai Chenshan Botanical Garden. Another sorry. Another linkage is from the section Elex. For this linkage is also have this ECP equine trait. Because most of this linkage is from the Tibet area in the very high elevation and very cold climate. Now I have found like this Crocus spinosa and Crocus semicapifolia have this acorn trait. And also even from the Tibet area, they also grows very well in our botanical garden because Shanghai is very hot. But outside in the outdoor area for the species from Tibet area grows very well. Um, the last one is from the North American area about these three sections. Uh, Now we just have one species from section Variantis. It's a very famous. It's the first species found this ECP acorn trait. This is the very famous individual in our botanical garden. It's a very beautiful landscape. And the second part is about the oaks collection based on our population genomics. Because now we know for each species there are a lot of different populations. Maybe these populations belongs to different have different population structure. They produce have special genetic characters. The first one we worked is about Crocus gilva. Based on our research, we found there are two very famous group for this species. The first group is the China group. The second group is the Japan and Southern Korea Jeju Island group. But from this map we can see the genomic structure is from the west to the east is they are scheduled changed from the pink color to the purple color. So we also collected the collected the seeds from different populations and planted some individual in our botanical garden. Except this, we also collected the seeds from different populations. The first part is from the Guizhou province and the west part of Hunan province. This links with the totally pink group. And then we collected some populations from the Jiangxi province with a small part of purple structure, and the other we also collected some populations from the East Sea line area like Zhejiang province and Fujian province with the more purple comments. Another species is about mountain cloud forest species, Crocus arbitifolia. For this species, there are only 7 populations. They always lived in the high mountain area. The elevation is higher than 1,000 meters. For this, from the population genomics, we can see the population from Guangxi province is very special, and and the population from the East China area have the similar genomic structure. For this species, we try to collect it as much as populations from the field, but it's difficult to find the seeds. We have focused on this species about 3 years. Up to now, we only found acorns from 2 populations, and only one acorn is collected from the Guangxi province, the special genomic structure, comments. Another species is about Cocos lithozoides. For this species, only lived in the Southern Sea area from the Shenzhen, Hong Kong, and until to the west part of the the west part of Guangxi province. There are only 6 populations for this species. Uh, Based on the population genomics, we found that there are three clusters for this species. So we collected all the seed because for this species, every year there are a lot of seeds. So we collected the seeds for these 3 clusters and planted in our botanical garden for for these 3 clusters. At the end, I want to show our oaks garden, the oaks garden in our botanical garden. The oaks garden was built in from 2020, just 5 years ago. A big area, behind our research center. There is a We designed the Oaks Garden as 2 parts. The first part is along the street. We based on the culture the Oaks culture because in the world there is a lot of country use the oaks as their national tree. So we designed this street as a national street and planted a lot of different foreign oak species. And inside this area, there are 3 small mountains, not mountains, small highland area. Because Shanghai is the end of Yangtze River, so just like 10 years ago, our government just wanted to protect the Yangtze River. So along the Yangtze River, there are more than 80 oak species. So we wanted to collect this oak species to plant it in these 3 small yellow cycle area. And up to now, in our oaks garden, we have more than 40 species. Like their national tree street, we have Cocos rubra, Cocos bicolor, Cocos ceres, and also Cocos nigra, pagoda, and comforter. In the we also collected some species for the inner area for the three yellow cycle area. For these three species from the Tibet area, the first one is Quercus lamellosa, and Quercus cuculensis, and Quercus spinosa. This is for the first the first sorry. This is for the first part of the oaks garden, and the second part is about the species. The native species lived in the second step of in China, like Shanxi province and Yunnan province and Guizhou province, like this area. Like these three species have planted in our botanical garden. Because the oaks garden is very new, just 5 years, so all the individuals and the trees are very small. Because for most of them, we planted from the seeds. So we prepared like 8 or 7 years ago and planted in the garden just 3 years ago. So all this is very small. And the last part is about some oaks in the East China area, like Quercus misifolia and Quercus anglana and Quercus glauca. Like this species, it, all these species are lived in the East China area. And now we also collected a lot of other acorns and to produce seedling in our lab, like the Crocus hui and Crocus champigny and Crocus flui and some other species. At the end, I want to give a short summary. Uh, According to our count, up to now, we have collected 62 oak species in our botanical garden. And we have conserved 3 species based on population genomics and genetic diversity. And our oaks garden has designed based on the oaks culture and our oaks research. Uh, Okay, that's all. Thank you for your attention. Um, I'm also searching for cooperation and exchange acorns. If anybody interested to exchange acorns with us, you can contact with me with my email. My email. Okay.
Great.
Okay, that's
Thank
all.
you.
Thank you.
Thank you, Yugang. That was fascinating. Really interesting research there. I was familiar with this, Quercus virginiana and the section virentis having that swollen root, but I didn't realize you had it also in those species in section ilex. Please, if you have any questions, feel free to unmute and speak up. We do have a few minutes for questions, and there are also some in the chat.
So I need to close my my share or stop my share?
Yeah, you can stop sharing. Yeah, that'll help everyone see.
Okay. Thank you.
You can see everyone. Well, if anyone else have a question, I'll say please do speak up. But I'll read one out from Jeroen, who's put a few in the chat. He says Quercus alnifolia also has a trait of germinating from the acorn scar. Does it have a special function? I can imagine they float on melting water from snow in the mountains so they don't sink. I don't know if you talked about germination from the scar as one of the traits you were looking at.
The germination.
Yeah, germination from the scar. Jeroen, if you want to unmute and elaborate,
Yeah,
please
yeah,
feel
yeah.
free.
I remember. Yeah, there are several species that germinated from the scar, not from the April, from the
Oh, from the tip.
especially for the species with the elongated cotyledonary petal. For most of this kind of germination style, they normally germinated from the scar area, not from the top.
Right. He also asks, I think in reference to your second consideration for collections, with different genetic patterns, do you mean that they can adapt to a different climate zone? Because I see a lot of differences between Chinese mainland specimen and a Taiwanese specimen.
Yes, for some linkages, they have special genomic components. Uh, with the landscape genomics study, we can see they have some adaptive genomics genetics to adapt different climate.
Okay.
Roderick, do you
Yes,
hear me?
go ahead. Yes, we do. Go ahead.
Well, I want to explain my Taiwanese specimen adapts much better here than my Chinese specimen. It's very, very slow, and the Taiwanese specimen is growing much better here. So that's really interesting to watch.
Are you referring to any species in particular or several?
I was referring to Quercus gilva,
Gilva only. Okay. Yeah.
which he mentioned in his presentation, because I already see so many differences in two specimens only from mainland China and Taiwan. So they have. I think the one from Taiwan definitely has a future here.
Great. Thank you. There's another question from Jason Yi. Why do many oak species native to China have difficulty producing viable seeds?
Why do many oak species native to China have difficulty produce variable seeds? No, most of the species can produce normal seeds. Just like the the species I showed in my presentation, Quercus abitifolia. I followed this species about 3 years. There are no seeds. I don't know why. I haven't researched on this. Just maybe just many years later they can produce a lot of seeds. For the other years, maybe there are just several seeds produced for each year. I think it's like this. But for other species, like the first species I showed, Cocos guava, every year you can see there on the big tree there are a lot of acorns produced. So
Can I
not all species of this like this.
contribute to that?
Yes, of course, Mick.
So one of the things we've discovered from looking at Quercus roba in England in detail is that there's a fantastic difference from individual tree to tree in terms of reproductive effort. So, for example, this year is a peak year for acorn production, but only on about 25% of the trees. An amazing 60% of the trees this year in a mast year have produced no acorns at all. And so it seems that there are some individuals, if you look at them long enough, just aren't interested in sex at all.
Thanks, Mick.
Well, Roderick, I want to do a little contribution about this as well. I see in the Netherlands, I see massive production of acorns, and year after year, it's just a mast year. I think it's caused by climate change as well. Trees are stressed, which it means they produce more and more acorns, and they drop early as well. They drop here by the dozens. It's very early in the season. Um, I see things changing rapidly because I'm in the field every day. Um, this is I think we have to watch this as well.
Thank you, Jeroen. That's an interesting observation. There was another question I wanted to share from the chat. Yeah, Sandra was saying, so informative, fab pictures. I notice all the different shapes of the acorns. Are there any ecological functions linked to these? So any ecological function linked to the specific shape of the acorns, Yigang?
Yes, the function. Raised. Yes, there are a lot of ecological functions linked to these different acorn trees. We just published one paper 2 weeks or 3 weeks ago, published in Plant Diversity. We just focus on the Quercus section Teclabalanopsis. From this research, we collected the data more than 10 years about the germination date, the size, the acorn size, and the position they germinate from the scar from the top. There we collected 6 different acorn trees to research the function of these acorn trees, the link of these acorn trees and with their climate characters, climatic factors, and we also building a new phylogeny tree to research on which is the ancestral traits, which is the devised traits, and which trait is better for speciation, and which trait is have more extinction like this. This is one of our new paper. Only working on the acorn traits, the link of acorn traits and their ecological functions, like this.
Great, thank you very much, Yigang. I think we'll close Yigang's presentation there. And before we end the webinar, I just wanted to open it up. Anyone have any questions in general about iOS business, about the meeting of members coming up, or any other topics? Looks like not. Well, I hope, as I say, we're able to make the meeting of members a virtual meeting as well as a in-person meeting. So I hope you can encourage you all to join if you're not able to be there in Oaxaca. And to close, I'd just like to thank our presenters again, Sandra and Yigang. Really fascinating presentations. I love the fact that we can hear from presenters from all over the globe and such a variety of different topics related to oaks. So thank you both for your time, for sharing your presentations, and thanks everyone for joining us today. And I hope to see you at the meeting of members, I say, or at the next webinar, which should happen sometime in November. Okay, thank you very much, and stay in touch and see you soon.
Thank you.
Thank you.